The core of it, it's about making tiny little droplets of water in oil. Okay, why would you do that? Well, the point is each one of those is like a tiny little reaction chamber, tiny little test tube, and you can make lots of them really quickly. You know, a million reaction chambers in a few minutes if you want to. So it gives you a really good way of doing big experiments fast. And the particular way we've applied it is into cells. Because once you have this idea of a little droplet of water, you can then put a cell inside that droplet, and then of course you can interrogate that cell, understand what it's doing, and on the basis of what it's doing, expressing or some phenotypic response, you can make decisions. That's an interesting cell, I want to continue with it, or actually that's not doing what I expected. So by having this idea of water in all droplets and putting cells in them, you can then start to build these very fast, very automated systems for looking at cells, making decisions about cells. And of course, that's a fundamental need within so many parts of life sciences. You know, it's all down at cells ultimately. And how do you understand what a cell's doing? How do you find the ones you really care about within often huge populations? So that's the core of it. One area in particular we're looking at is functional assays. So a lot of our work up to now has tended to be a single cell, say, secreting or expressing a molecule, and you're looking for that in some way. What we're now moving towards is what we call co-encapsulation. So rather than having a single cell, you would deliberately put two different cells into a droplet. And then of course you can start to look at the interactions. You know, how do those two cells work together? How are they communicating? How is one influencing the other? Particularly if you're looking into applications like cell therapy, for example. If you're looking to engineer a cell to have some impact on another cell, you can then start looking at those interactions. So that's a really interesting piece of extension of the core idea to go to that sort of functional assay level. A second thing we're working on quite a lot recently is actually starting to bring gene editing technologies into the droplets. And that's doing the editing process in the droplet, not doing a bunch of edits and then dropletizing, it's actually doing it in the droplet. So we've been looking at both transduction and transfection methods for doing that and getting some really interesting results, that you can put a cell in a droplet, make genetic change to that cell, and then look at the consequences and decide if that was useful or not, which opens up a whole new way of potentially screening big libraries or doing those kind of genetic modifications on cells. So Cyto-mine is really taking the droplet technology and turning it into a product that people can use. You know, it's great talking about droplets and what you might do with them, but you've actually got to make it usable. We also just sort of clarify the reason we call it picodroplets. I mean, why pico? It's just a sense of scale. They are actually at picoliter volumes. That's why we call them picodroplets. But no, the Cyto-mine is a specific example of using droplets, picodroplets, for an application. And that one's really targeted at things like antibody discovery and cell line development. So it has quite a specific workflow built into it in terms of you put cells in, it encapsulates those cells, so it does the single cell per droplet. It will then incubate those to allow them to express, say it's an antibody, you know, they will start to express the antibody. The Cyto-mine then has lasers, so you can actually start to look at fluorescent signals, and that lets it do sorting. It will actually say, you know, this cell is expressed, we'll hold on to it, that one's not really done anything, will not. So you then are able to sort those cells, and then finally it's able to dispense them back into the traditional well plate format, which of course is extremely useful for moving on to the next step of the process. So what Cyto-mine does is it brings all those different pieces of the process all into one box with the consumable, so it's much more hands-off for the users. You know, they don't have to worry about all these droplets and process flows and things. It's much more from their perspective. I put a population of cells in, I know the assay, I run the process, and as an output I get a subpopulation which I know are high quality, high producers, really useful to me. So that's the core need for a Cyto-mine. It's taking the technology and putting it in the hands of users in a much more usable, automated way, and as I say, particularly for things like antibody discovery. And it has a huge impact because you really can screen an awful lot of cells, you know, millions of cells in an 8-hour day, pretty much hands-off. The picodroplet technology, there are so many places you can see it starting to go. As I said earlier, something like a Cyto-mine is much more around antibodies and cell lines, but that's just because where we're starting. You know, when you start to look out into things like non-mammalian systems, yeasts, bacteria, the whole world of industrial biotech and synthetic biology which is growing. That's something we're very much starting to look into, particularly with some of the mass spectrometry work that we're doing. If you start to look into things like cell therapies, we talk about the functional assays, you can clearly see where that would go. If we start to look into things like stem cells, neural cells, there are so many potential applications. Now, it gets harder because you're putting cells into droplets. Some cell types are kind of more naturally comfortable in that environment than others. So there's a lot of work to do, but you can certainly see how the underlying technology could be applied to many areas which maybe are more researched today, but certainly in the next 5, 10, 15 years, no, they will be coming to market and becoming new, genuine, products and capabilities. And so it's some of those areas that we're exploring to say, well, how do we bring this underlying Picodroplet technology, apply it to these research areas so that we are able to, again, take that step change in capability, that what might just be a, well, it'd be lovely if we could do it type conversation becomes a, yeah, here you can do it. More than that, here's a thing to do it with, and we know it'll work.